So you are welcome. You are welcome to my next lecture. A series of lectures on bioinorganic chemistry to deal with the structure and function of metallo-biomolecules. This lecture, I'm concentrating on copper-based biomolecular devices for dioxygen activation. My last lecture was concerned with biomolecular devices for dioxygen activation, but they are all based on iron. I told you all the biomolecular devices for dioxygen activation are based upon either copper or iron. So in the previous lecture, I talked about two types of iron-containing biomolecules. One is porphyrin-based iron complexes, iron metallobiomolecules. The other one is non-heme iron devices, which activate dioxygen so that you'll be able to oxidize at normal temperature and pressure, normal pH, organic compounds to their hydroxylated derivatives. So again, I shall show you here the cartoon which shows the reduction of carbon dioxide with water, hydrogen atoms of water, where you get the carbohydrates and oxygen is the byproduct. So in the reverse reaction, dioxygen, which is a byproduct of this photosynthesis, will be used in respiration, or in general they will be used in oxidizing the organic compounds, so that in the presence of catalysts, so that you get back the energy. So this energy will be used to drive all the reactions which happen in living systems. So these employ catalysts. So I was able to tell you in my last lecture how these catalysts can be, iron biomolecular devices or copper-based biomolecular devices. So I told you how the oxygen is activated by these complexes, and how in some cases it is possible to activate the substrate itself by dioxygen. So the spin multiplicity problem was there, that can be overcome by supplying energy so that you will get a triplet oxygen. So the next you should also remember that the dioxygen that we obtained, that we make use of in this dioxygen activation, will be from the decomposition of water, where you generate 4 electrons. You will see today how copper-containing biomolecular devices make use of molecular oxygen again to oxidize organic compounds. And the reactions, the overall reaction that we are going to see will be the reverse of this one. That is, oxygen will be reduced by electrons either to water or hydrogen peroxide. So we shall look at some what novel biomolecular devices which are able to activate molecular oxygen. So I also consider that this is an occasion for me to tell you how metals are dealt with in bioinorganic chemistry. When metals are present in our body, for example, is very high in concentration, that is 1%, they are called bulk elements, or they are also called macronutrients or bulk electrolytes. They are nothing but the pre-transition metals, that is alkali cations and alkaline earth cations. And when the metal ions are very small in amount, catalytic amount, so we have the transition metals. This is the important point I want to emphasize. That is, what we deal with are the copper and iron containing metal enzymes. So they are present in very small quantities, trace quantities. So they are made whenever we need to do some molecular oxygen activation. So we have dealt with already the iron containing biomolecular devices. Today, as I said, we will look at the copper-containing biomolecular devices. So this is just metal content of a human body. So the amount of copper and the iron that are present in our body are very small, 4.2 gram. Copper is 72 milligram, right? Whereas the alkali metal ions are very high in concentration. So we just look at the role of iron in biology. This is what I talked about: dioxygen transport, storage, and also the oxidase enzymes, electron transfer and nitrogen fixation. So when you have less than what is needed, the amount of iron needed for our body, we get anemia. And also we get a lot of disorders of the immune system. The same way when copper is involved, it's a part of the metal ion, or sometimes it is a cofactor of the enzymes. They are called cofactors of the enzyme. They will be involved in electron transfer and dioxygen transport and oxidase activity. So this is what I talked about during my last lecture, that is dioxygen transport by copper-containing hemocyanin. The oxidase reactions will be looked at. So in absence of required amount of copper, we will have weakness in the artery, and also that will be giving liver disorders, right? So again, I want to repeat how metabiomolecules are dealt with, that is proteins, and then when they are involved in these reactions, in these functions, they are called transport proteins. When they are involved in chemical transformations, they are all enzymes, right? So enzymes can be divided into you know um, several types based on the reactions. We have already looked at the iron-containing metallo-biomolecules, which are involved in very interesting chemical transformation. Today, I shall take up these copper enzymes. So they also are involved in dopamine hydroxylase reactions. They are involved in oxidase reactions, that is complete oxidation of the substrates. We just today we will take up some examples from this list and then talk about it. We have copper containing nitrate reductase. We have copper zinc containing superoxide dismutases. So we take up these enzymes one by one. Catechol oxidase, galactose oxidase, and ascorbate oxidase. In fact, our own research in earlier research in Bharathidasan University Chemistry Department involved galactose oxidase enzyme modeling. And we should also remember what are all the functions of this copper. This is the electron transport function which we looked at already at length, and metal storage, how metallothioneins are used to store copper. We have not looked into that. If we have some time, we will look into how copper ions are stored in nature. So this is what we looked at, dioxygen transport we looked at. So today we will take up dioxygen activation. How dioxygen is activated by copper-containing metallo-biomolecules. Before that, I should tell you a very interesting thing, that is how many types of copper proteins are there. Already we have looked into two types of copper: type 1 copper, type 3 copper. Type 1 copper correspond to you know um, blue copper centers. They are involved in electron transfer. This is what we spoke at length, how copper becomes suitable for reversible electron transfer. I told you how the structures, the active site structures of these proteins are very abnormal. Abnormal in the sense they don't have square planar geometry, which is normal. So they have non-planar distorted geometries, and they have 3 plus 1 coordination. They have, when the structures are normal, then the spectral properties will also be abnormal. So we have blue color corresponding with this charge transfer transition. We have DD transition, but at very low energies, because the geometry is almost tetrahedral or low coordinated one. So they will be observed around 6,000 waves per centimeter, whereas Square planar compounds will have their ED transition, ligand field transition around 600 nanometers or 20,000 waves per centimeter. These proteins have a very characteristic signature, that is, they have very low A parallel value. In contrast to this, we have type 2 proteins. What are these type 2 proteins? They are all non-blue proteins. They don't have a blue color like this. That means we don't have mercaptide to copper to charge transfer. That means cysteine amino acid is not involved in coordination. More than that, the structures are very normal. Normal in the sense they will be having a square planar geometry, almost square planar geometry, normal geometry. They will be distorted ones. Since they have normal structure, they will have a normal spectral behavior. You know that aqueous copper compound or cuprammonium complex will have a DD transition around 625 nanometer, that is 20,000 waves per centimeter, and the epsilon will be very low, 12, 50, like that. Same way here also, the epsilon value, the ease of excitation of this ligand field transition will be less than 1000, right? In that sense, they will be normal. They have a normal spectra. So the EPR spectra will also be normal. The G parallel, will be around will be in the range 2.200 to 2.400. There is an wide variation. That's again another interesting aspect about EPR spectroscopy as applied to these type 2 copper proteins, right? So you can notice that based on the structures, the copper proteins can be classified into type 1 and type 2 structures. That is based on the active site structures. So copper proteins can be classified based on either structure or based on the spectral properties. Blue copper proteins, I can say, are those which have very low A parallel value. Whereas type 2 proteins are those proteins which have EPR, which have active sites with higher A parallel value. That means around 142, 200 gas. So they have normal EPR spectra. So they can be, copper proteins can in nature can be classified into two types. And their function is O2 activation, activating the dioxygen. Dioxygen, as I told you, has a double bond. So they can be made reactive by coordinating the dioxygen to copper. That is by decreasing the bond order, right? So that is the secret behind the type of chemistry we are going to discuss about the copper protein. When the copper proteins are involved in oxygen activation, that means these are mostly copper enzymes. They bring about chemical transformations involving molecular oxygen. Then we have the type 3 proteins. Already I talked about it, type 3 protein. So they will have an abnormal, that's not an abnormal, that's having a dimeric structure, copper-copper structure. Now you catch the point that this type of copper-copper interaction is there in hemocyanin, which is involved in dioxygen uptake, right? So dioxygen uptake is performed by hemocyanin. So hemocyanin is a type 3 copper protein. It is not involved in any chemical transformation. So type 3 are simply copper proteins. So they are characterized by the O2 minus, the oxyhemocyanin. Proteins are characterized by this type of charge transfer, O2 minus 2 copper 2, observed around 600 nanometer. So the most important thing is that these type 3 proteins, though they have copper 2 copper 2, they don't exhibit an EPR signature. They don't give rise to an EPR signal because, so they are all EPR inactive. So what are type 3 copper proteins? They are not type 2 and type 1, but they are EPR inactive proteins or EPR silent proteins. So based on the EPR spectroscopy, you can divide all the proteins into three types. These type 3 proteins can be divided further into three types also: type A, type 2A, type 2B, type 2C. More than that, I would like to tell you that there are copper enzymes. They are called multi-copper enzymes, multi-copper proteins, right? They have more than one type of active site for carrying out the dioxygen activation. They have type 1 site also. They have type 3 site also. So all of them, or most of them, are called multi-site copper enzymes. So copper enzymes are a little bit complicated in the sense all these types are involved. This means that the type 1 protein involved takes part in electron transfer, delivers electron for the O2 activation, catalyzed by type 2 protein, and type 3 also it takes part. Now you can simply say that the O2 needed for the copper for the activation and the chemical reaction, chemical transformation is uh, held up by O2, right? Dioxygen is taken up by type 3 and stored there, so that can be now used by type 2 protein. So type 1 protein delivers electrons, type 3 proteins provide dioxygen for dioxygen activation. So a cooperation among all these three types are needed. When you have more than one type like this, three types, we have a new type of copper protein. You need not have a new type, but conveniently we will say a type 4 protein. So there are number of type 2 proteins and then type 4 proteins. Now you understand the important point that you have copper proteins to perform not only electron transfer function, it is also there to do dioxygen activation function. So this is a very interesting topic. That is how copper proteins are classified into several classes. That's very interesting, really. So we have seen actually what are type 1 proteins. They have a tetrahedral, almost tetrahedral geometry, two imidisoles, one thioether, one methyl
Hello, sir.
coordinatively saturated the site. So it is this four two. Hello?
Hello.
Did சொல்லுங்க.
சார் இந்த இது ஸ்லைடு போட்டிங்கல்ல இந்த ஃபர்ஸ்ட் படம் வைக்கும்போது கொஞ்சம் ஸ்டக் ஆயிடுச்சு சார்.
இந்த படம் இந்த இதா?
ஆமா சார்.
ஸ்டக் ஆயிடுச்சா? மறுபடியும் ஆன் ஆயிடுச்சா?
இது வந்து வந்துருச்சு சார்.
மறுபடியும் போயிட்டு வரணுமா?
ஓகே சார்.
இல்ல மறுபடியும் பிரசன்ட் போய் வரணுமா?
இல்ல இங்க இருந்தே ஆரம்பிக்கிறேன் சார்.
Good, very good, very good. Okay. So we have type 1 proteins and we have type 2 proteins, which are different from the type 1 proteins in that they have a square planar geometry. Again, you can see imidazole, histidines or histidyl imidazoles are bound to copper to stabilize copper in this plus 2 oxidation state. So you have a normal geometry, and the fourth position is occupied by water molecule. This water molecule can be easily substituted by your substrates, right, which are needed to be oxidized, right? So that will be substituted by your substrate, Another important point is the geometry is a distorted one because that is needed to allow the binding of substrates here. Look into detail how these different types of copper proteins, copper enzymes are there to activate the molecular oxygen, right? You have another, the third type. This is what I was telling you, hemocyanin, third type. You have imidazole, right? Three imidazoles on one copper, three imidazoles on another copper. And some of the type 3 proteins will have a link here, OH minus link. It has not been yet characterized in several proteins. In the case of hemocyanin, also it may be there, but there we looked into how this is replaced by, this group is replaced by dioxygen at the in between the two copper ions. So these are all dimeric copper. So when copper 2 and this copper and copper interact, the unpaired electrons are paired and you get EPR silent spectra, EPR spectra. So these are the essential differences. So I think now you will understand the fact that the enzyme that we are going to talk about will make use of electrons from this site whenever needed, or they can store their oxygen at this site and use it for dioxygen activities. So I mentioned about type 4 copper site that is there in ascorbate oxidase. I told you it will have more than one active sites. It has a copper site, copper 1 site, type 1 copper site. See, histidine, water like that. I'm sorry, this is type 2 center. It's not having type 1. It doesn't need any electrons for the reaction, conversion of ascorbic acid to ascorbate, dihydroascorbate. So this is a type 2 site. So type 2 site is found in association with a type 3 site, right? But they are at non-bonded distance, at a distance. So when copper 2 in ascorbic acid oxidase is involved in enzymatic function of oxidizing ascorbic acid to dihydroascorbic acid, it needs dioxygen. So it cannot hold dioxygen. See, it has only one vacant site, two histidine and other sites. So you cannot directly take dioxygen and then transfer to the substrate here, substrate which will be bound here. So we have a support from in nature by the type 3 site. So dioxygen will be bound in between these two copper. See, it's very wonderful that how nature has designed its active site so that the dioxygen bound here can be utilized by copper, which is bound to the substrate here. So you have a substrate enzyme complex like this. So an oxidized ascorbic acid will look like this. See, 3.4 amps a minute. And when it is reduced from tricopper site, reduced site, they are all present in copper 1 oxidation state. And see, this distance is increased. This is a hydroxide ion I talked about, X minus, which links these two copper. So we have two forms of ascorbate oxidase which are involved in the binding. So anyway, I will spend some time towards the end of my lecture on how this ascorbic acid is involved in activating the molecular oxygen. This just tell you that the type 4 site can be considered as type 2, a mixture of type 2 and an assembly of type 2 and type 3 site. But conveniently, we can also say they are all multi-copper oxidases, that's type 4. This is what I was telling you, the EPR spectra of copper proteins. Castocyanin has a very simple EPR spectrum where the A parallel is very low. It's all very much compressed. So the A parallel value is 70 gas or 60 gas. This is the prostaglandin one, prostaglandin EPR spectrum. Here is an ascorbic acid oxidase spectrum. So what about this spectrum? This will be having, I told you this is having a normal copper 2, right? Normal copper 2 geometry. So in that case, you will have a normal EPR spectrum, normal EPR spectrum, right? With a high A parallel value. It will be higher than 70, probably towards 140 Gauss or so, higher A parallel value. Because this spectrum has been condensed, it may appear that they look similar, but they are not. So they will have a high A parallel value, not low A parallel value. In addition to this, you have another important signal here, right? So these signals, the G parallel in between these two and the G perpendicular here, they are characteristic of square planar copper, axial copper. In addition to this, you also have another weak signal here. And these signals are correspond delta ms is equal to plus or minus 2. While these G parallel and G perpendicular come from delta ms is equal to plus or minus 1 transition, EPR transition. This signal, which is very weak in intensity compared to this one, see? So this arises from the copper-copper interaction. This copper-copper interaction gives rise to, this is delta ms equal to plus or minus 2 signal. So this is very much characteristic of the presence of type 2, both type 2 and type 3 copper in the enzyme. So multi-copper oxidases will have more than one type of copper which can be diagnosed with the help of the EPR spectrum like this. So in addition to normal EPR spectrum, you will have a very weak signal like this. So you should be able to observe for these proteins at very high gain of the EPR machine, right? So these are again some more EPR spectra of pantothionin, type 1 protein, stellocyanin protein. See here also you have some unresolved band, unresolved apiral signals. So this is more or less rhombic. We would always say that sometimes this signal will be inverted. Here, let's not go deeper into this one. So we may have four signal, apiral signal here and the signal here, stellocyanin. So they are all having very novel geometries and against novel EPR spectroscopy. Plant scientists, biologists they may not know exactly what is how these signals arise, but they use these signals to immediately characterize and classify the copper proteins that we have in plant systems and also in bacterial or animal systems. So these protein signals spectra are very important for you if at all you want to understand more about copper in biology. So there I told you about this type 1 protein. They are all mononuclear. There is only one copper in it. They have unusual properties. They are involved in plant photosynthesis, bacterial photosynthesis, azurin. Type 2 proteins. What are type 2 proteins? They are mononuclear copper synthesis. So the donor atoms like imidazole are from protein residues, imidazoles. And you have only one water molecule in galactose oxidase. A good example is dopamine beta hydroxylase. And the side chain oxidation of dopamine, that is alkyl CH group, is oxidized and hence corresponding you have dopamine to norepinephrine. This is very important transformation to maintain the nervous system, right? And the type 2b proteins will be having donors from protein side chains like the imidazoles, and in addition you will have some unusual protein side chains. This is the most interesting aspect. They are unusual protein side chains. These protein side chains will have amino acids which are modified amino acids, derivatized amino acids. So whenever you need the enzyme, these proteins will be made and they will be assembled. Okay, so we have very interesting aspects like this in the proteins we are going to deal with. So galactose oxidase is a good example to demonstrate this one, this idea. That is, protein side chains are derivatized. They are involved in oxidation of alcohol in fungi. Right? Alcohol is oxidized to the aldehydes. Types 2C. What are type 2C? They are also having copper in the normal geometry, normal properties. But in addition to this, you will have another metal, dinuclear metal. So in addition to copper, you will have zinc also. So you will have copper zinc superoxide dismutase, whose duty is to just degrade degrade superoxide, dismute them, that is decompose them, copper zinc superoxide. So in addition to this, zinc will be there. But then, though you have a zinc, this is classified as type 2C because the copper has a normal geometry, normal EPR spectrum, normal redox properties, and all other things. So we shall try to look into some basic aspects of this in due course. The type 3 proteins, as I was telling in hemocyanin here, they are dinuclear. So copper-copper interaction will be there, antiferromagnetic interaction. So you have EPR silent species, hemocyanin, which I talked about at length in my last lecture, involved in oxygen transport. Another enzyme is tyrosinase enzyme. Orthohydroxylation of phenols, right? To get dihydroxyphenols. So they are all very interesting enzymes, which I shall take up in due course. So they all have dinuclear copper site, right? They have dinuclear copper site to carry out this function. Tyrosinase uh, mm, will have an ability to convert, not only phenol but other substrates also. So type 4 proteins are trinuclear. This is what is said in ascorbic acid. It's a good example, right? In addition to ascorbate oxidase enzyme, we also have a very interesting lactase enzyme. Lactase is involved in oxidation of polyphenols and polyamines in plants. This lactase has very good biotechnological applications. So it is essential for bioinorganic chemists to know about the active site of lactase and try to understand whether you can generate, synthetic mimics for all these compounds. I told you how synthetic mimics can be obtained for type 1, and to some extent you can get synthetic mimics for the type 2, but it is very difficult to get synthetic mimics to have these geometries, dinuclear geometry or trinuclear active sites, very difficult. So in that case, one should first understand this one. Probably in future you will be able to make a model for lactase. So galactose oxidase is a type 2 enzyme. As I told you, we dealt with this enzyme supported by Department of Science and Technology. We tried to make models for this. So when we took up the work around 89-90, there was hardly any understanding of this enzyme. So this any enzyme is an extracellular enzyme secreted by the fungus, namely Datylium dendritis. Right? They catalyze the 2-electron oxidation, not 1-electron oxidation, 2-electron oxidation of primary alcohols by O2, dioxygen. So alcohols are oxidized by dioxygen to aldehyde stage. See, these enzymes are all very specific. You cannot oxidize this one to the acid, carboxylic acid, with the help of this galactose oxidase. And see, the interesting aspect is you get hydrogen peroxide. So one of the oxygen atoms of dioxygen in the air is converted, is donated to, is transferred to alcohol like this, and the other is given to water. The other is given to water, and you get hydrogen peroxide. So this is very interesting one. And more interesting thing is the how do they bring about this specific oxidation. See, this involves, I said, two electrons, but this enzyme has only one copper center. So you may wonder, two copper ions may be involved to deliver two electrons for the oxidation. No, these enzymes make use of only one copper, and another electron needed for the activity. Enzyme activity comes from the coordinated ligand, namely the polypeptide. So that electron is present always as a free electron. So these enzymes are called free radical enzymes. So they contain one unpaired electron on copper, another one on the enzyme organic matrix itself. So these two can interact several ways. So both the oxidation reaction here, reduction are two electron redox processes, right? So I told you already in my last lecture that all enzymatic reactions involve pairs of electrons. Okay. So here is the enzyme. Here is the enzyme active site, enzyme structure, galactose oxidase. This enzyme active structure was published in 91 or so, and we published our model in 92 when we communicated our results to Dalton Transactions. The referees asked me to look into the latest paper in Nature where the structure has been published, and told us about the novel property, the novel type of bonding we came across in our model. Right? So we published it as a communication in Dalton Transactions. Right? So what is the active site structure for this copper present here? So this copper has a very distorted square planar geometry, right? You have imidazole again. Look at that, two imidazole. So they are all ubiquitous in biology, in all our biological metallobiomolecules. And we have a phenolate in the axial direction. See, that is bound to copper at a very long distance. Remember, copper is a Jahn-Teller ion. So this axial ligand cannot approach copper very closely. So that is bound at a very long distance. What is this amino acid? Tyrosine amino acid. The phenolate oxygen is bound to this, the axial direction. So you should remember that the phenolate is a strong field ligand. Phenolate PHO minus, strong field. But then it would be expected to coordinate in the equatorial position. But the enzyme matrix, the organic matrix, compels the phenolate to coordinate in the axial direction. More or less, the active site is pre-organized, right? So phenolate, though it's a strong field ligand, it occupies the weak Jahn-Teller axial direction. Look at this one. Another phenolate is involved in the equatorial plane. So this galactose oxidase enzyme contains another tyrosinate, but it is involved in the equatorial position, strong bond. Look at that. This particular moiety, phenolate moiety, the tyrosine phenolate moiety is different from this. This is a normal tyrosine side chain. It is contained in side chain. Now what is this? So this is what we call it as, a modified amino acid. This alpha position here has been taken up by cysteine sulfur. So the protein itself makes use of one of its cysteine to come here make a covalent bond. When you need the enzyme for the oxidation reactions, it is not synthesized and stored in the cell. They are all assembled, right? At this point. So, how this is assembled, how this is derivatized itself is a very important bioinorganic chemistry problem. So, that we call it as genesis of the enzyme. So, whether the genesis of the enzyme itself involves the same copper. So, you have umpteen number of questions, interesting questions around this type of enzymes. Okay, so these enzymes are novel. I told you this copper is having one unpaired electron. So where is the other unpaired electron residing? The other unpaired electron is residing on the phenolate here. So the phenolate bound here is actually existing as a radical. So this unpaired electron and this unpaired electron have every possibility to be paired. So always such enzymes will be, when you isolate it and store it, they will be interacting, that will be inactive. So what people use to do, they use this extract, okay, galactose oxidase extract, treat it with potassium ferricyanide so that you can oxidize this and then you get the free radical. So please note that this is a free radical enzyme, right? Which has different oxidizing capacities, oxidation levels, we say, right? So two electrons are involved in the oxidation of aldehyde to alcohol. So that is supplied by not to two coppers ions, each with one unpaired electron, so that you get two unpaired electrons. But you get this oxygen to have unpaired electron. You may wonder how this unpaired electron is stabilized. That itself is again another interesting aspect. This unpaired electron is delocalized into the benzene ring, right? The delocalized electron on this phenolate is again stabilized by the formation of a donor acceptor complex with a tryptophan moiety present somewhere here. Tryptophan moiety of the protein comes below this particular entity, moiety, and then stabilize and then makes a pi bond bonding at 4 angstrom unit distance. So this particular pi interaction serves to stabilize delocalize the unpaired electron and then stabilize it. So the free radical enzyme is stabilized by a very novel type of donor acceptor complex. This is a donor, tryptophan will be an acceptor. There are several proteins where tryptophans are used uh, take up electrons or stabilize some excited states. So these are the novel properties of the enzyme, right? Let us see how the enzyme activity works. So this is the copper protein active site. Your phenolate radical here, unpaired electron is here. This phenolate, tyrosine, phenolate, amino acid phenolate is here, right, like this. So what is this? This is your alcohol substrate. You know, alcohol substrate has an oxygen atom with two lone pairs. So this substrate can bind to, can donate its electrons to copper 2 by replacing water. So the first step is substitution of the water molecule by the substrate. So this is nothing but an enzyme substrate complex, right? So when you have substrates like this, see, what you happens is this unpaired electron, this H here, this H here, see this H plus, since it is strongly bound to this one, this proton will be easily knocked off. So this bond is polarized, knocked off. So how does this knocked off proton is absorbed? Do you need a base? No, you already have a base here, phenolate minus. So this proton is transferred to the same active side. This becomes O minus. So this bond is very weak. That is why it functions as a base. So that is why this bond is broken. So it is protonated. See, it's very interesting that you have a very interesting internal base available to deprotonate the protons, right? So absorption takes place when this becomes OH minus. OH. So this bond will be broken, right? A phenyl is protonated, so this bond will be broken. So during these electron rearrangements will take place, so you get an aldehyde. So alcohol here will be oxidized to aldehyde stage. So the O2 that we make use of, right, is used to convert this to aldehyde. So O2, where is O2 now? O2 now is bound at this point as a superoxide radical to copper 2. See, this is vacant. So this is vacant. So this is occupied by superoxide ion. So O2 minus O2 gets an electron, right? And then binds to this. You get hydroperoxy type of bonding is there. So this one is undergoing again scission. Bond undergoes scission, and then you get hydrogen peroxide, right? Hydrogen peroxide is liberated. So the oxygen that we supply is used to bind to copper directly and get activated because you have vacant site generated here, right? So that is activated, and you get the rearrangements. Excuse me. So you get this type of oxidation, right? So if you just look at this one, this is a two electron system. So start from this free radical. You have one electron added to this tyrosine oxygen. See, another electron added to copper. First electron to this, second electron to copper 2. So you get this system. The reverse direction, this electron is removed from copper, right? It's copper 1. So this one is broken, right? Almost tetrahedral, towards tetrahedral geometry, low coordination 1. So that is why copper 1, you know very well, like iron 2 has an affinity for O2, copper 1 will have an affinity for O2. That is why how copper 1 is able to bind to molecular oxygen at this point. So, so electron is taken away from the dioxygen here, bound here. Another electron is from copper. Another electron is taken up from the oxygen here, the bound oxygen here. So it involves two levels. There is two electrons, or in two electrons. A pair of electrons is involved in the oxidation reactions. So this is the novel, uh, you know, uh, uh, enzyme activity. So what is that we did? So we use this tripodal ligand. I told you when Kitajima was involved in making tyrandate, tripodal ligands to make copper compounds. I was using in our laboratory tetradendate ligands, but they are tripodal, topology. So when we can try to change these uh, donor sites, see, pyridine by imidazole, by different type of compounds like this, substituents like this. We also made compounds of this type, two compounds like this, two moieties with these two hydroxyl groups. So we made very novel ligands. This work was done by one Dr. Uma, was from Gandhigram Rural University at that time, 90. Now I believe he is the principal of Lakshmi College of Education. So she made very novel compounds, copper compounds, and she was the person to, for the first time, to make a model compound with the phenolate oxygen, right? So this is the compounds crystallized around 90 or 90 or so. We should took some time to publish it in 92 as a communication. We felt that it's very interesting phenolate in the axial direction. At that time, this crystal structure was not of this character oxidase was not known. So we were well excited that we have a structure like this, phenolate in the axial direction. See? So as a simple coordination chemist, we try to publish it. What is this one? This is an acetate. So this is the acetate. So the enzyme also you can just compare. These are the ligand which we used. Two pyridine moieties and then one phenolate here, nitro or phenolate. So this is the active site I told you, right? So this has almost the same type of active geometry, very much resembling like this phenolate here. Of course, we have only another heterocyclic nitrogen instead of a phenolate here. So you have water. Since you have water in the liquid solution, we got an acetate. This again is very interesting. We're able to make more number of compounds, a large number of compounds, and we're able to publish these results in inorganic chemistry in 98. So you can change this bond, this ligands. It can be water, chloride, azide. You can change this anion. And when you change the anion, when you have a strong donor like here, you can very well understand that this bond will become weaker. That means this phenolate will act as a better base. So you can very well see that the Yanteller ion principle tells us that by changing the anion, the copper oxygen distance can be changed and the basic property of the phenolate can be changed in the enzyme. So that means this, this particular enzyme activity should depend on the anions present here. The anions present here. In fact, the proteins are isolated as an anionic complexes. So depending on the bound anion, the activity will be different. This bond distance will be different. In fact, and we also found that the depending on the anion, this bond distance varied. So through our systematic study on the modeling system, we established that Yan-Teller ion, a normal copper to copper geometry, unlike your type 1 copper, is essential to make this phenolate act as a base, internal base. So now we understand the important differences between blue copper type 1 active site and this particular active site. So we need normal copper to bind to substrates. So water will be replaced, acetate will be replaced by our alcohol. That time we didn't have much facility. We just were able to say that when alcohol was added to this, it was activated to aldehyde. That's what we were able to prove, but we didn't, progress further in that one. And when I presented this work in Germany for the first time in our bioinorganic chemistry conference in 1995, everybody was excited to look at my poster. Then there started lot of activity on free radical enzyme, that is galactose oxidase enzyme. How many galactose oxidase enzymes are there? There are umpteen number of galactose oxidase enzymes are there. Very many enzymes were based on this one was published, and all of them have the type 2 copper site. So now you understand how nature has designed a type 2 active site, a pre-organized active site, so that you can use this site to bind to alcohol and partially oxidize it to the aldehyde, right? So we made use of a number of techniques, EPR, everything, to characterize this particular complex, and then we came out with the conclusion that they have normal properties. The property that we obtained compared very well with the spectral properties of the enzymes. So we published a number of papers on this in 1998, 92, and also in later on in 2000. So we were very successful in making such a model, but we couldn't do further work on this before I undertook the got some facilities to do these oxidation reactions. A lot of people abroad, my counterparts in other universities like Sheffield, University of Sheffield, and the bioinorganic chemistry Max Planck Institute, which I visited to deliver a lecture on this ion catechol dioxygenases. They became interested in this one, and they found out very easily that these synthetic mimics can convert alcohol to aldehyde. So the second oxidase reaction, oxidase enzyme is copper amine oxidase. These copper containing amine oxidases are ubiquitous enzymes. They are in bacteria, fungi, plants, and animals. In fact, the human vascular adhesion protein, which deactivates some cellular function and which are which lead to cancer. These copper these proteins have been shown to be, which is simply called as VAP1 protein, was shown to be copper amino oxidase enzyme. So this is an inhibitor enzyme, right? So you can prevent cancer activity with the help of this enzyme. So if you make a model for copper amino oxidase, you can use it as to block some of the reactions in cells to prevent the formation of the cancer, right? So this VAP1 is implicated. This regulates glucoside trafficking and glucose transport, and it is implicated in, I'm sorry, the amino oxidase is implicated in glucose transport, implicated in diabetes mellitus, congestive heart failure, and liver diseases. So making a model is very important. Not only understand the wonderful things about the amino acids, which I will describe now. This has inspired the synthesis of therapeutic agents as inhibitors. That's what I told you here. A lot of organic chemists are involved in this amine oxidation. A lot of activities there. Inorganic chemists were also became active by a group from the uh, Max Planck Bioinorganic Chemistry Center. Um, they also became interested in this copper amine oxidases. What is copper amine oxidase? That catalyzes the aerobic oxidation of amines to imines. That is NH2 groups to imines containing C double bond N. Oxidation, right? CNH2 C double bond NH, right? Sorry, C double bond N. Amines to imines. So this is the most important reaction, industrially important, and it's also important from the point of view of synthetic organic chemists. Okay? And this protein is, again, not a simple enzyme. This is a protein where we have a copper center. See, type 2 copper center. So you have um, imidazole again, right? Another imidazole, another imidazole. There are 3 imidazoles bound to copper. See, one, this is the active site geometry. 1, 2, 3. And you have one water molecule here in the axial direction, the copper. There's also another water molecule involved. It's an approximate geometry. So this is, almost a square pyramidal geometry, not tetrahedral geometry. And the most important thing is this particular active site is having a neighbor. That neighbor is called a quinoprotein, having a quinoprotein moiety. See this water molecule here, one of the water molecules here, the longer distance, is bound to the uh, cofactor that is called the quinoprotein cofactor. So that contains both copper as well as the topoquinone cofactor, TPQ, this one. That is your dioxygen, which is the oxygen is bound to the hydrogen atoms of water, another double bond here. Okay, so this is a very interesting molecule, and we also try to work on the mechanism. It's very difficult to understand the mechanism. Still, light has not been thrown on the function of copper and this topoquinone. This type of topoquinones moieties have been synthesized by organic chemists in 15 steps and use that as a cofactor to bind to amines and make imines, right? So This topoquinone itself is very interesting cofactor. A lot of organic chemistry has been developed around this particular topoquinone, and this how these topoquinones were made. People believe there is a mechanism by which this particular moiety, you notice that topoquinone, I'll just show you topoquinone moiety is co-assembled with this center. This is derived from tyrosine. I'm sorry, I'm sorry, sir. It's derived from an amine. So even this amine itself is oxidized by this copper, converted to quinone. So the very part of this active site is, generated by this presence of copper. This is a theory, but there is no demonstration of the proof for this. And so the assembly of this enzyme, this whole active site itself is very interesting. We have questioned whether copper is needed for this one, oxidation of these amines to aldehydes and then to ketones, right? And during my stay in IITB during 1914 to 16 as a distinguished visiting professor to the Department of Chemistry, we had a very interesting student working with Professor Murugavel. Since I was associated with his group to some extent, one of the students came up with a very interesting compound. So Murugavel they tried to illustrate how the compound was formed. They couldn't solve this. And once when I entered into his room one day, he just turned around and then said, "Here is Palani, why don't you just show your result to him?" And that result, when we worked on further, through light on this aspect, copper is needed to make this particular topoquinone. That is the so-called chicken and egg problem, right? How this chicken was created from this, how this needs copper again. So that type of activity is called off and on mechanism. That we threw light on this particular mechanism, we found that such topoquinone moieties can be generated with copper. So the the novel compound isolated by the student, through light on not only this one, the topoquinone moieties he made was able to catalyze at ambient temperature, ambient conditions, ambient air, oxygen present in air is sufficient to do this oxidation, right? So this we found to be very good for as a catalyst for oxidation of amines to imines, right? So we had a very interesting results. One is this particular topoquinone is formed catalytically because organic chemists have used 15 steps to generate the topoquinone, but we generated it in one step. So not only that, we use this metal complex as a catalyst, wonderful catalyst, which brings about 100% transformation of amines to imines. That's what we demonstrated in the laboratory of Professor Mergovel. So this is the most interesting discovery we made. The genesis of the active site, the assembly of the active site was illustrated, and we also generated a very good catalyst for the amine to imine transformation. This is what I've been telling you, topoquinone. So the enzyme is an oxidoreductase enzyme that oxidizes this is the tyramine. The tyramine. This will form the basis of our topoquinone, etc. So when they catalyze the aerobic oxidation of primary amines to aldehydes, these aldehydes will condense with the primary amines. See, primary amine to form 0,1N bond. See, this will condense with another mole of this amine to form aldehyde. So also we can notice that hydrogen peroxide is generated. Remember what I talked about earlier, this oxygen evolving complex. That is the decomposition of water to give dioxygen. Here I told you the reverse of that reaction. That is where dioxide is reduced to get hydrogen peroxide. That is also important. So various amine substances were used as sources of uh, carbon and nitrogen. So this is also very important to maintain the carbon spent here. So this carbon cycle is also connected to this amino oxidase. So that inspired the development of several biomimetic and homogeneous catalytic systems. So this is the TPQ in the CuAO enzyme. See, just imagine this type of cofactor. Generation of the cofactor is difficult. People who have made this type of TBHPQ, they have got very interesting results. Published their papers in Nature that they were able to convert with this moiety alone. They were able to demonstrate that for different substrate scope, they were able to bring about a reaction normal temperature. Anti number of catalysts because it's very industrially important. People worked on this, published a lot of papers in uh, Nature, at least by this larger one by in France. She was able to publish a number of papers in Nature. So what we got is that different types of TBQ has been isolated. And a large number of papers have been published by organic chemists, very large number, thousands papers have been published making use of this type of cofactor, organic catalyst. Okay, this can be regarded as an organic catalyst. Okay, when there's a small amount of this combined with amines, different amines in bulk quantities, and then use this. But they are all very unstable at times. People who have got, they've also used copper oxide or something as a co-catalyst. That points to the fact that copper is essential for this type of enzymatic reaction. And here is the model we got in IITB. See, you have topoquinone, a very complex one, in fact. So this is the model generated. This nitrogen here was bound to copper. So this was crystallized as soon as the research scholar mixed the ligand with copper immediately. Within half an hour, this was formed. So very fast, very pure form. 99% of this catalyst was generated containing copper. So both copper and this topoquinone moiety were assembled together in one particular catalyst. That is the biggest discovery which I consider as very important uh, in my career also. So this type of was made, and we found that the very copper complex carried out this amine to imine conversion at room temperature, whereas all these compounds used higher temperatures. Enzymes use low temperature, 25 degrees Celsius, or biological temperature, right, 33, and pH, very low pH, 7.3 or so, and we were able to do that, imitate every aspect of the copper amine oxidase in this one. So I was insisting with Professor um, uh, uh, that we should be able to publish this work in Nature. But he wanted to complete all the study and then publish, so that delayed the publication. So we were able to get this particular molecule having a tetrahedral geometry, right? So that we published it not in Nature, but we were able to publish it in another journal, that is ACS journal, ACS uh, catalysis, which has a very high impact factor like 15, right? So this is the amine. What is the genesis of the catalyst? This is the amine made by used by the student, right? Okay. So she condenses this amine with salicylaldehyde, a swift base, and she prepared the copper compound. So when she exposed this copper compound, carried out the function at methanol at room temperature, she got this particular copper compound. See, just see what is that you got. You have two diethyl groups, and see, you have two diethyl groups here. But in the product, the two diethyl groups have moved here. So that is the reason why the people there was Murugavel's group, they were unable because he's not basically, he's an inorganic chemist, well recognized, celebrated inorganic chemist, but they were not having good bioinorganic chemistry. That's not bioinorganic chemistry laboratory. So when they sold this compound to me, I got the idea and sat down in the overnight and then worked out the mechanism for the formation of this particular ligand. So it's not a decomposition ligand, but this involves a very interesting rearrangement, right? That's what we found, and that rearrangement involved the genesis of the catalyst, where superoxide is used by the very copper itself here. Copper bound with molecular oxygen, converted into superoxide, and sent it to this particular point, and then this rearrangement happened. So this is the catalytic activity, which I'm trying to point out. Alkyl amine, primary amines, right? In acetonitrile O2 stream, 1 mole percent of the catalyst, we got this. When we get the aldehyde, that will condense with another mole of the amine to get these amines. 100% conversion, 100% selectivity as in the enzyme. We can also use cross coupling like this. See, cross amines. This is having CH2, this doesn't have CH2. For this also, there was activity. Though there was 100% conversion, 100% selectivity, people using organic catalysts have failed in getting these cross-coupled compounds, but we were able to show that this reaction is catalyst is very efficient, right? So this, in fact, one should study, go through our paper to know how this one-to shift occurs. So this we were able to publish in ACS Catalysis, an unprecedented copper. Sridhambara is the student who made this compound, stumbled upon this compound by accident, right? So I was associated with Professor Murugavel in this work, and Gopal and Rajaraman made some computations on these molecules. He was his associate professor there, IITB. He was our own former MSc student, so we all collaborated, and we had a very good publication in ACS catalysis, right? So we have copper enzyme, copper oxidase enzyme, imitated by a synthetic metal complex. Not only we mimicked the active site, and we also mimicked the enzyme activity, catalytic activity. We were also able to demonstrate that copper is very much essential to generate the topoisomerase cofactor, and it is also very much essential to further catalyze not only the genesis but also in the use of the copper 2 compound. In the absence of copper, the catalytic activity will be less, but in the presence of copper 2, we found the catalytic activity is enhanced. Within half an hour, the catalytic activity will be over. The catalytic function will be over. And we were able to demonstrate that this copper 2 also assists in the catalysis in the amine oxidase activity. So we were able to demonstrate very well that both copper and the cofactor are involved in catalysis. In fact, it is this Largeron, after looking into our paper, wrote to me actually that we should be able to collaborate further on these types of copper amine oxidase enzymes. So the other type 2 copper enzyme is superoxide dismutase. This itself is textbook aspect for MSc chemistry, bioinorganic people who learn bioinorganic chemistry. It's a copper zinc superoxide dismutase, antioxidant. It's an antioxidant, remember that. Because that is able to decompose the superoxide radical. I told you superoxide radical formation is the terminal reaction in bronze in our respiration reactions. So if it is not removed, this will oxidize um, many types of cell damage. So to prevent this superoxide dismutate works to decompose this and convert it not to water but to hydrogen peroxide. So these reactions are very interesting. That's why I take time to illustrate each and every enzyme and then illustrate the mechanisms. So these are all present in plants, aerobic organisms, as well as in plants, right? So this superoxide dismutase is located in mitochondria, found in nucleus, as well as in cytoplasm, right? So it's used as an antioxidant. So in human, it is located in cytoplasm and mitochondria, right? This has been subjected to a lot of work. This is the enzyme site. Okay, you have I think dimer also, two copper ions, copper and zinc are there, right? Two active sites are there. I don't want to do much details. So the active site structure contains this particular ion, copper ion and zinc ion. What is that you see? This is a normal copper, and you have a water coordinated. So all these four nitrogens are bound very strongly, and zinc is here. What is this? This 3-imidazole is also imidazole, but then this N lone pair is donated to copper, right? And this nitrogen, imidazole nitrogen, has been deprotonated. Pyrrole hydrogen has been deprotonated. So that is occupied by zinc. Again, this is supported by, is stabilized by histidine side chain amino acids. So it is a very complex, metal complex. So imidazole functions as a bridging ligand, bridging copper and zinc. The role of zinc is still questionable, but we would always say that this moiety is there to support copper too. This takes part in the enzyme activity, replacement of water by your superoxide moiety, and then rearrangement, right? And electron transfer from superoxide minus to copper, so you get copper 1. When it is copper 1, you know very well it will favor tetrahedral geometry, so this bond will be broken. So this moiety doesn't go away from the active site because it is the protein moiety which keeps this reorganized like this. So this is decomposed, broken, and the active site, is undergoes this copper one donates electrons again, and then deactivates copper. 2 moles of superoxides are deactivated by this system. So people have been investigating the role of copper and zinc. So we say the zinc is just not only reactive support, provides reactive support, it also supports to maintain unusual geometry on. So the function of zinc is manifold. In fact, we generated with tripod ligands multinuclear copper zinc heterodinuclear active sites is Dr. Uma, which I was mentioning. She generated a lot of such metal complexes, but we couldn't publish them all. So superoxide binds to copper too. See, look at this one. Superoxide binds to copper center. So this copper imidazole bond here is broken. This bond is broken. I told you, get a copper hydrogen. This one is used as a base to have the proton. Copper 1 is there. So copper 1 reacts with superoxide, superoxide from the respiration. So again, it transfers electron to this. So it gets converted into copper 2 again. Immediately the imidazole bridges here. You get by the copper 2 imidazole active site. So this alpha electron on delivered to this one. So you get hydrogen peroxide and water. In the presence of proton, that conversion happens. So just calculate the reverse reaction H2O plus H2O plus 4 electrons to give 1 mole of hydrogen peroxide like this. So this copper 2 irreducible zinc superoxide can also interact like this. I think I written it again directly where you get water, right? So this is how the secret behind the role of zinc, how it functions as to maintain the broken, keep that site inactive, and how it scavenges the superoxide and prevents the cell to undergo any damage. And in fact, people relate the active superoxide activity to the lifespan of people. You know that lifespan of human beings are controlled by these superoxides, right? Let me now turn over to type 3 enzymes and also enzymes, multi-copper oxidases, where you have more than one type of site. So we have tyrosinase enzyme. That's very much essential in our own human body, right, to make the dopa, right, the drug for keeping the nerves active, tyrosinase enzyme. So copper oxidases are ubiquitous in nature. They utilize and activate dioxygen during catalysis. This is what we saw already. That is copperamine oxidase and then galactose oxidase. They are involved in electron transfer or oxidation of organic substrate. When you have type 4, more than one type of copper sites, you can see that electron transfer is also involved. So the function of tyrosinase is to introduce one hydroxyl group here, hydroxylation from oxygen atom of the air what O2% in water in the atmosphere, right? The same reaction can be brought about by catechol oxidase. There is another copper-containing enzyme type, 2, type 3 enzyme, and type 3 and these two type 3 enzymes, they also differ to some extent. They are involved in the same type of, conversion. So type 3 copper oxidases. Your type 4 copper oxidase also does more or less the same type of reaction, that is lacase. Another one is ascorbate oxidase. So that contains not only this type 2, 3 side to carry out this oxygen activation, dioxygen activation, and incorporation of one of the oxygen atoms into the substrate. No, that has more than type 3. It also contains type 3. It involves type 1 and type 2 also. So it's a very complex enzyme, lacase enzyme. It's very difficult to reproduce the enzyme activity or make models for this, because we have to assemble 3 such sites together in one place. So this is how copper oxidases are important. So this is catechol oxidase. So this is a very famous enzyme in modern times. Almost all copper chemists would have touched upon this one, because you make any copper compound that will try to oxidize catechol to benzoquinone. In catechol oxidase is involved in living systems as well as in plants. Convert this catechol to orthobenzoquinone like this, right? And then again convert them into melanin. So this melanin formation is the basis of, um, you know, browning of fruits. When you cut an apple, for example, it will be very bright. After some time, it will turn into browns. That is because of the formation of the activation of catechol oxidase enzyme, the presence of the dioxygen when you expose it to molecular oxygen, convert it to benzoquinone and then react with amines and convert it into some tricyclic melanin, polymeric melanin. That is also responsible for our brown color, melanin. So plants also have this type of catechol oxidase. So what is that they do? They try to introduce one of convert the oxidation reaction like this. So in this reaction, you can see O2 is reduced by electrons to give water. One of the oxygen atom is involved, the other oxygen is involved in reacting with H plus to get water. So this is I said the reverse reaction. The reverse reaction is nothing but oxygen evolving complex. So this is of fundamental importance in biology. So here, look at the active site. Very briefly, I will tell you that this is the active site of oxycatechol oxidase. Deoxy1 will have no dioxygen. Again, we have the work done by the contribution from Kitajima used here to talk about the active site structure of oxycatechol oxidase. Or this is the intermediate involved in the catechol oxidase mechanism. Okay, so these copper atoms are antiferromagnetic coupled, so this is copper inactive. So EPR inactive one. So oxyform is this one, deoxyform, you will not have dioxygen. So how does this function? How is this able to bind to molecular oxygen and use this oxygen, bound oxygen, to be transferred to this carbon? How does it do? Here is a simple mechanism. So this is the structure. Copper A site, copper B site. Although it would appear both of them have the same active site, there are differences. 3 imidazole here, 3 imidazole here. Right? This is the enzyme structure, some different way of representing the dinuclear copper site here. So this is the oxyform, this is the deoxyform. So both the copper atoms are linked by a hydroxyl group. Almost you see that this is the structure very similar to our hemocyanin, right? But this one is used to activate molecular oxygen so that this oxygen atom will be utilized for CH activation, whereas the other oxygen, the other copper site in hemocyanin is used to store molecular oxygen, right? So it's wonderful that nature has chosen dinuclear copper site, not only for dioxygen storage and transport, also for dioxygen activation. So type 2 sites, mononuclear copper sites, are involved in oxygen activation. This dinuclear copper is also involved in activation of molecular oxygen. That's a wonderful thing. So what is the mechanism? You always start from this particular inactive form, deoxy form, copper 2A form, copper 2B form, B copper, they're bridged by hydroxyl group. So what do you do? You start from here, interact with catechol. How does it interact? It interacts on one copper. Probably I think this has a weakly coordinated water molecule. That water molecule is substituted by hydroxyl group. Substitution, ligand substitution. So the other hydroxyl group is oriented towards this oxygen like this. So now what happens? You get the quinone. I'm sorry, I think this is a very simplified form. I will just anyway illustrate. I should have one more slide to illustrate this one. So when you have, copper one obtained here, this is oxidized into quinone, and in between these two, you have now copper one form. This copper one has an attraction for molecular oxygen. Okay, so that binds with another mole of catechol and one mole of oxygen. See, it binds another mole like this. Dioxin is formed here. So this is oriented towards this copper 2 side. So this is oxygen atom bound to this. So this hydrogen interacts with this weak oxygen atom because already this has interacted with oxygen, another 2 more copper 2. So hydrogen bonds with this one and deprotonated, and that is how takes place here also, right? So this is how this deprotonated and it is converted into quinone. Okay, so it's catechol oxidation mechanism. The mechanism involves both the copper dinuclear mechanism. The other enzyme I talked about is H-response. Okay. So tyrosine is also, I told you, it's very similar to, the reaction is very similar to a catechol, but there are all differences. So present in animal and plant tissue, this also does the oxidation, monophenol to orthophenol oxidation, like your catechol oxidase. In addition to that, it also mediates the orthohydroxylation of phenols. That's called phenolase activity. So tyrosinase, catalyzes not only orthogonal formation but also the CH activation to get orthohydroxylation. A phenol can be converted into catechol. So both the tyrosinase and the catechol are called, phenolase active enzymes. They are all called polyphenol oxidase enzymes, right? So how though these two have more or less similar active sites, there is essentially a difference between tyrosinase and then catechol oxidase. So that is done by incorporation of a glutamic acid in tyrosinase active site. Catechol, catechol oxidase doesn't have this glutamate, so um, I'm sorry, wait. So you have asparagine also involved. By mutation, you introduce an asparagine. So tyrosinase differs from catechol oxidase by the presence of asparagine. Okay, so that asparagine combined with glutamate in catechol oxidase, you get tyrosinase, right? And also, as I said, there is a water molecule, conserved water molecule, known as the copper, to abstract a proton from the monophenol. So they are both are the same, but they are all obtained by mutation. So tyrosinase can be site directed. So this enzyme can be converted into catechol. Catechol can be converted into this one by simple mutagenesis of an amino acid. Both are having more or less the same. It's the asparagine in tyrosinase. There is no asparagine here, right? So they differ. When this is introduced, it has additional activity, that is mediation of orthohydroxylation of phenol. Right? So this is the active site, dinuclear active site in tyrosinase. So what's its duty? It serves. It's also involved in conversion of tyrosine to dopa. How? This is tyrosine amino acid. So that is converted into it undergoes converted into dopa by a series of reactions. What does it do? It just converts hydroxylation of this one. See, you have introduced hydroxylation. L-dopa is obtained. So tyrosinase converts L-tyrosine amino acid into L-dopa. Hydroxylate this one. Mediates. Tyrosinate mediates this one. So the next step is, this there's another enzyme which will introduce hydrox. I told you, this will hydroxylate the aliphatic carbon also here, right? So that leads to formation of dopa. Is L-dopa, and that corresponds. You get the drug, which is very important for our nervous system, right? So how is it done? Mechanism. See, this is the phenolate. Right, there is a histidine side by side. Binds one of the copper like this. Three histidines, three histidines. This is oxytyrosinase, right? So this is this CH bond is oriented towards the oxygen like this. So you have more or less the transfer of this oxygen, activated molecular oxygen, activated oxygen atom from oxygen atom from activated molecular oxygen. See, that is introduced here, and you have a quinone, right? So this is the dope I talked about. So that is how dinuclear copper is useful in activating molecular oxygen like this, binding and activating and transferring it to the carbon atom here, ortho hydroxylation. So this is also um, enzyme catalyst is corresponds to CO also, catechol oxidase. Both these can do this type of oxidation, but you need tyrosine specifically to do this reaction of converting tyrosine into this. This type of compound can be done like this. See, when you get tyrosine, this can do both functions, but this can do only one function. CO cannot do this particular hydroxylation. No phenolase activity. This is called phenolase activity that cannot be uh, carried out by a catechol oxidase. So ascorbate oxidase is the next multi-copper oxidase. The active site contains, I told you, a type 2 protein, type 2 center, and a type 3 center. Now that I have told you about these enzymes, oxidases, you can now easily figure out that this should be copper 2 site, right? To do the enzymic activity, and this copper site, hydroxide bridge of histidine. So this is present in other enzymes also. So this is essential to keep your dioxygen here, activate that, and transfer the oxygen to this copper site. So this is a copper 2 site, type 2 site. A parallel is greater than 140, greater than 70. That means its type 2 center, located far away from this type 3 center. Function is catalyze the catalysis of the oxidation of L-ascorbic acid to dihydroascorbic acid. There are umpteen number of good inorganic complexes also which are able to bring about ascorbic acid oxidation. It is interesting to look at this particular reaction. So here also 2 electrons are generated. Those 2 electrons can be handled by them easily. So the enzyme is blue-green in color. It turns yellow when a small amount of ascorbic acid is added. Substrate is added. The yellow color changes back to blue upon exposure to dioxide. So this is the enzyme activity. You can also take up banana, cut them open, add some citric acid, and then show the brown color to your students and children how it is browny. It establishes the presence of this particular tyrosinase enzyme, right? And you can also expose it dioxin and see how enzyme is acting. If you want to prevent your apple from getting brownies, and as soon as you cut it, just put it in salt water, and you just prevent it from coming into contact with molecular oxygen. So this enzyme exhibits an absorption band at 606 nanometer, high epsilon, and the source a solder at 412 nanometer, high epsilon. So it is these two are responsible for the yellow color. This is assigned to catechol in the ascorbate to copper to charge transfer because it is very intense and disappears as soon as you add ascorbic acid. That sits on the ascorbic acid sits on copper 2, replaces this one. So reduces copper 2 to copper 1. So in that case, you have no charge transfer band. ASC minus to copper 2. LMCT band, ligand to metal charge transfer band will not be there. So this just tells you the spectral properties and the ascorbate oxidase mechanism. As I pass on to lacase, a multi-copper enzyme, right? This is the enzyme which contains 4 sites. What does it do? It converts 4 units of COH to C double bond. So 4 electrons are generated. Again, the accompanying reaction is this one, O2. For this O2 is employed. I think I have not indicated this here. And this removal of hydrogen, you get this. So this O2 is now used to abstract these electrons, 4 electrons, and convert it into water. The reverse of oxygen evolving complex reaction. These conversions are very complex, and this is a multi-copper enzyme. For all the 3 types of active sites are present here. So lacase, what is it? It's an oxidase enzyme involved in the oxidation of polyphenols and polyamines. It's a typical example to illustrate the roles of different copper active sites. Electron transfer, activation, storage of molecular oxygen, activation of molecular oxygen. So all of them are involved in oxidation of the substrates using dioxygen. As a byproduct of oxidation of phenols, in the presence of air is water. See, the byproduct is water. So you are able to remove these electrons, but that is absorbed by molecular oxygen, you get water. So the byproduct is water. So this enzyme is a harmless enzyme. So this is used by people in organic synthesis, in biotechnology, in textile industry to decolorize the pulp, paper industries, and for food applications also to bioremediation as well. So this is very important. That's why I took it up at the end so that you will try to understand the chemistry behind this and also appreciate the role of lacase. Now, what are these sites? It's a fungal lacase. So the type 1 copper is type 1 copper is here, stitched into this site, complex enzyme. So what is its role? It takes up electrons. See, you need electrons for this reaction, right? Electrons for this reaction to activate oxygen, molecular oxygen, to convert superoxide like that. So copper one side, imidazole, the sulfur, cysteine sulfur, fungal lacase. At 12 angstrom 13 angstrom separated from type 3 copper. So type 3 copper is dimeric copper, imidazole, hydroxylated, hydroxy bridged. Copper, you have a lot of space. So molecular oxygen can be captured here. So that molecular oxygen captures here and receives an unpaired electron from this copper. See, becomes O2 minus and transfers to this type 2 copper as in galactose oxidase, as in ascorbate oxidase. So this is a trinuclear cluster put together, trinuclear cluster. So this binds to molecular oxygen and then activates it, converts the COH groups to C-double-bond O groups. So that is how the oxidation of your compounds or substrates are conducted by these ones. So this is how we postulate how type 1 copper site obtains the electrons, delivered to type 3 site to copper, and then you get the conversion of O2 to water. So this is a multi-copper enzyme. Okay, so this is how we can illustrate the role of all oxidases and how they can function as biomolecular devices to transport molecular oxygen, to bind molecular oxygen, transport molecular oxygen, and activate molecular oxygen, and also involved in synthesis of hydroxylated aromatic compounds, right? So just remember it is the copper 2, copper 1 redox, which is underlying the function of all these oxidases, where there is complete oxidation. Remember, instead of we also have a number of other copper enzymes, but some of the enzymes which I dealt with are dealt with are from our own experience. I thought I would concentrate on these oxidases, not on other type of oxidases. So more number of copper enzymes and proteins are there, so they involve in more types of biological functions. So thank you for hearing me. Okay, so we shall go take up in the next lecture type 6 some newer type of things, inorganic biochemistry type of thing, that is interaction of metal complexes with DNA. That's another activity which we pioneered in India. So I thought I would deal with this type of topic in my next lecture. Okay, thank you for your patience.